water is fundamental medium of life, making up 70% or more of the total weight of most of the living organisms. Because water molecules are polar, they are highly effective solvent of hydrophilic substances, including sugars, nucleic acid and salts. But water is not merely passive backdrop. It's also slightly ionized, a chemical behavior that underpins the entire concept of pH. In an aqueous solution, water molecule continues exchange proton with each other. When a positively charged hydrogen nucleus or proton dissociate from one water molecule, it is immediately hydrated by adjacent water molecule to form hydronium ions, leaving behind a hydroxyl or hydroxide ion. This rapid transfer is facilitated by process called proton hopping, where proton hop between the hydrogen bonded water molecules, explaining why acid-base reactions in water are exceptionally fast. By convention, we often refer to hydronium ion concentration simply as the hydrogen ion or proton concentration represented as H positive. At 25 degrees Celsius, this reversible ionization reaches an equilibrium governed by an equilibrium constant. Because the concentration of pure water is vast and constant, 55.5 moles, we multiply it by equilibrium constant to define the ionic product of water, designated as Kw. At 25 degrees Celsius, Kw is always equal to 1.0 into 10 to the power 14 molar. In precisely neutral solution, the concentration of hydrogen ions exactly equals the concentration of hydroxide ion, meaning both are present at 10 to the power minus 7 moles. To avoid working with highly widely scientific notation, we use logarithmic pH scale, where pH is mathematically defined as negative logarithm of hydrogen ion concentration. Because the pH scale is logarithmic, each single unit change on the pH scale represents tenfold difference in the hydrogen ion concentration. For example, a cup of black coffee with pH 5.0 has 100 times more proton than pure water at pH 7. Meanwhile, a common carbonated cola with pH of 3.0 or glass of red wine with pH 3.7 has hydrogen ion concentration approximately 10,000 times greater than that of human blood, which is slightly basic at pH 7.4. When different substances are dissolved in water, they function either acid or bases, shifting this delicate ionic balance according to the Bronsted-Lowry definition. An acid is proton donor and base is proton acceptor. Strong acids such as hydrochloric acid dissociate completely in dilute aqueous solution, releasing their proton easily. Strong bases like sodium hydroxide also completely ionize, producing hydroxide ion that combines directly with the hydrogen ion to form water, thereby driving down proton concentration. In contrast, the substances most critical to biological systems are weak acid and weak bases, which do not dissociate completely when they dissolve in water. A weak acid, such as acetic acid, exists in irreversible equilibrium with its corresponding proton acceptor, known as the conjugate base, which in this case is acetate ion. The characteristic tendency of any weak acid to lose proton is defined by its acid dissociation constant, or its negative algorithm. A lower pKa value indicates stronger weak acid that dissociate its proton more readily. For example, an amino acid like glycine possesses a carboxyl group with pKa of 2.34 and amino acid with pKa 9.6. The carboxylic group of glycine over 100 times more acidic than standard acetic acid, which has an average carboxyl pKa of 4.76. This dramatic downward perturbation of pKa in glycine is caused by its chemical environment. Specifically, the electrostatic attraction of the nearby positively charged amino group, which withdraws electron from the carboxyl group and facilitates the release of the proton. To maintain a stable internal environment, living organisms rely on biological buffers, which are mixture of weak acid and their conjugate bases that tend to resist change in pH when small amount of acid or base are added. If you titrate a weak acid with strong base and plot a resulting pH against the equivalent of base added, you obtain a titration curve. The midpoint of the titration, exactly half of the weak acid has dissociated, making a concentration of the proton donor equal to that of the proton acceptor. At this unique midpoint, the pH of the solution is numerically equal to the apparent pKa of the weak acid. The shape of this curve and its buffering action are described by the Henderson-Hasselbalch equation. This equation reveals that a conjugate base pairs is an effective buffer within zone extending approximately 1 pH unit on either side of pKa. Within this buffering region, added hydrogen ions are neutralized by reacting with the conjugate base, while added hydroxide ions are neutralized by proton releasing by the weak acid, minimizing the shift of pH. In the human body, keeping pH tightly regulated is matter of survival, as conditions that push blood pH outside the narrow range of 7.3 to 7.5 are life-threatening. Human blood plasma is normally maintained at pH of approximately 7.4. This constant physiological pH is achieved largely by bicarbonate buffer system, which is complex. Coupled equilibrium involves carbon dioxide gases in lungs, dissolved aqueous carbon dioxide in blood, carbonic acid and bicarbonate ions. In our tissue, the CO2 produced by the oxidation of organic fuel in mitochondria is hydrated to form carbonic acid, a reaction catalyzed by enzyme carbonic anhydrase. Carbonic acid then dissociates into protons and bicarbonate ions. The pH of this system is governed by the ratio of bicarbonate to carbonic acid, which in turn is equilibrium with partial pressure of carbon dioxide in the lungs. If excess protons enter the blood, such as lactic acid, from vigorous exercises, the reaction proceeds towards the formation of more carbonic acid, dissolved carbon dioxide, which is then exhaled by the lungs because we can adjust our respiration rate to quickly change the carbon dioxide concentration in our lung. This system acts as a highly dynamic buffer. However, if individual hyperventilates due to stress, they breathe out too much carbon dioxide, which raises the blood pH to 7.4 or higher, causing alkalosis. Conversely, several clinical conditions can result in acidosis. In untreated diabetes mellitus, the lack of insulin forces the body tissues to use stored fatty acids as their primary fuel. This metabolic shift results in the rapid accumulation of high concentration of 2-carboxylic acid, beta-hydroxybutyric acid, and acetoacetic acid. The dissociation of this acid lowers the pH of the blood plasma below 7.35, causing diabetic acidosis. At this lower pH, crucial metabolic enzymes do not function optimally, leading to headache, drowsiness, stupor, or potentially life-threatening comas. Why are enzymes and other proteins so incredibly sensitive to these pH changes? The answer lies in the relationship between pH, protein conformation, and electrostatic interactions. Proteins are long chain of amino acid linked by peptide bond, which folds into precise stable three-dimensional shape. This folded state or conformation is stabilized by thousands of weak non-covalent interactions, such as hydrogen bonds and electrostatic attractions. Many amino acids have ionizable side chains, such as basic side chain of lysine, arginine, and histidine, or acidic side chains of aspartate and glutamate. The electrical charge carried out by this ionizable side chain is entirely determined by the pH of the surrounding medium. Among these, histidine is specially unique because its side chain's imidazole group has pKa of 6.0, allowing it to easily transition between protonated and unprotonated forms near neutral pH. This property allows histidine residue to act as a highly versatile proton donors and proton acceptors, facilitating many enzymes catalyze reactions. From the microscopic proton hopping that make water reaction lightning fast to global carbon cycle buffering our oceans, pH is the chemical that dials shape of all biological life. It is a masterclass is how small simple ions control the grand structure of life. If you found this deep dive into chemistry of pH helpful, please like, share and subscribe to Life Science.